All right, I am recording. So do you guys have questions? Where do you want to start?
I don't necessarily have anything that I'm specifically struggling on. I just came here because I knew I definitely needed like an overview of what I should be focusing on and remembering to like go over.
Okay. Anybody else? It'd be helpful to have a direction. We've been through 100 and something PowerPoints. So I'm not going to be able to start at the beginning and go over them. So
I just tried typing. I'm not a good speller. So I was asking if I wasn't really the greatest with chemistry. So I was asking if we could do like some chemistry review.
all right, good place to start. Let me share my screen. Got to get used to being back on this screen. I don't think that's what I want to share. Hang on. There we go. Try that again. Share screen. There we go. Okay. So you can see the sodium potassium pump right now, right?
Yes.
All right. I'm just finding chemistry and where chem starts. All right, Taylor, are you good with the basics of an atom? Okay.
Yeah.
So you're okay with that.
Yeah, mine was more like with the movement through like cells and the double membrane and that stuff.
So the energy part at the end?
Yes. Yes.
Oh, at the end. At the end. Gotcha. All right. So let's see. Where am I here? So we talk about cell energy. Let's see. Let's get on this one. This is a good place to start. So when we talk about cell energy, our ultimate goal in the cell is ATP. And what we always say, and you hear it all the time, we need glucose, right? We need glucose. And we do need glucose, but glucose is only the beginning stage. Glucose is only what gets into the cell and gives us the ability to make energy, because the cell itself doesn't use glucose. The cell, in order to make things run, is going to use ATP. So this is the ultimate goal. And we saw with the ATP that it mostly came from the electron transport chain. So glycolysis is going to break down the glucose into 2-pyruvate. That's the goal of glycolysis. The other thing about glycolysis is that makes it different is it's in the cytoplasm. Okay, so glycolysis is broken down in the cytoplasm. So we talk about ATP production, and most all ATP production is going to be in the mitochondria. The small exception here, right, is where we get a little bit of ATP from our breakdown of glucose. So if I were to ask you, for example, where is most of the ATP produced, you would know that that was in the mitochondria, right? Because the electron transport chain produces a lot more than glycolysis. This I told you didn't need to worry about. I was just pointing out in this where we use up glucose. We actually have to use glucose, or not, ATP. I don't know why I'm having such a hard time with that today, but ATP, we have to use ATP to get ATP. And after glycolysis, everything moves into the mitochondria. And the Krebs cycle is first. And the Krebs cycle also produces 2 ATP. So it also produces a little bit of ATP, of cellular energy, but not very much. But it, in the process, produces 10 coenzymes, reduced, because they have electrons, reduced coenzymes. So they are carrying electrons. Once something is carrying the electrons, then the next step is to dump it off, right? It's to dump off those electrons. And those electrons are all going to get dumped off at the electron transport chain. That is also in the mitochondria. And if we actually had to, which you won't have to, but you could go back and you could look at each of those NADHs. And for each NADH, each enzyme or electron carrier, you could calculate how many ATP come from that. So for each NADH, the electrons are going to produce about 3 molecules of ATP. And for every FADH that is produced during the Krebs cycle, about 2 ATP are produced. And so that's the power of those electrons. And remember that, and this actually explains why the FADH only has 2, but the NADH makes 3 ATP. And that's because they come into this and drop off their electrons in different locations, if you wondered why. But, and then look, once it loses its electrons, it's oxidized. So this is the oxidized form. This is the reduced form. So this is dropping off its electrons, and now it's oxidized and it can go back and be reused and pick up more electrons. This is where oxygen comes in. This is why we breathe oxygen. It's why oxygen is necessary to us to survive, because the electron transport chain requires oxygen to pick up the electrons that have gone through it. And if there's no oxygen present there, then the electron transport chain stops. But we get most of our energy, our ATP, from the electron transport chain. If we were to stop this because we didn't have oxygen to make this reaction work and end up with water, then we can't make that 32 ATP. We can't live very long. ATP is actually a very short-lived molecule. It's not around for long. It's not a stable molecule. And because it's not stable, it's not going to hang around for long periods of time. So after about 5 minutes without oxygen for us, we see irreversible consequences to our tissues. And that's because we've stopped this reaction, which is stopped energy production. The actual production of ATP is coming through this windmill. So because there's a lot of hydrogens on this side, and there's not many hydrogens on this side, our gradient, our diffusion goes from high concentration to low concentration, and it comes through this molecule here, this protein in our membrane. And by doing that, that gives us what we need for energy to put a phosphate on an ADP to make ATP. Okay, so that's how we end up getting all of that ATP from the electron transport chain. That gives us our summary, where we have 2 ATPs from glycolysis, and 2 ATPs from the Krebs cycle. And then we have 32 from our electron transport chain. Good, Taylor? Do you need to go back through any of it? I'm fine going back through it if you,
No, I think I got it.
okay. And then we just talked about the importance of oxygen. And we talked about lactic acid, right? Building up lactic acid when we don't have the oxygen here. So we end up doing what's called anaerobic respiration. And it's anaerobic because it's without oxygen. Aerobic respiration is what we just saw. All the parts, the glycolysis, the Krebs cycle, and the electron transport chain are all part of aerobic respiration. Once oxygen is taken away, it becomes anaerobic respiration. And that's where we see the lactic acid build up.
Can we
Taylor,
go back
is
to
that-
the summary
Just a
page? real
second.
quick?
Hang on one second, Katie. Taylor, is that a dog or a cat or your feet?
That's a cat or a dog.
Oh, okay. Go ahead, Katie. Sorry.
Okay, I was just admiring the dog for a second. It's so cute. I was wondering if we could go back to the summary page real quick.
That one?
Yes. Thank you.
You're welcome. Got a whole bunch of other stuff. How do we know we're alive? What are our characteristics of living things?
Carbon.
From a chemical standpoint, what about when we started out, our characteristics of all living things? Remember that?
Our cells?
Cells are one. Yep.
DNA.
Okay. DNA. You've got to grow and reproduce, and that requires DNA. Homeostasis, right? Every cell tries to maintain homeostasis. Now we're multicellular and we have ways to do it, but every cell, even down to a little bacterium, wants to have homeostasis, that internal steady state. They require energy. They respond to changes in their environment. And populations evolve. We don't talk much in this class about evolution, but our DNA changes over time, over generations, and that's what leads to evolution. That's led us to be from being all one common ancestry cell to being all different types of cells, and multicellular versus single cellular. I'm not sure that's the right word, but so that's our 6. We have them down as 6 characteristics of life. What makes us human? What are the characteristics that make us human?
23 chromosomes.
Actually, we have 46 chromosomes.
Yeah, that's what I meant.
The terms and
What?
what is it? I can say it. The terms. That's what I don't
Yeah.
know.
Opposable thumbs, right?
I know our cells don't have walls. I don't know if that
Yeah,
qualifies.
you're right about that, but that doesn't separate us from other animals, because our other animals don't have cell walls either. But you're right, that is definitely something that will tell the difference between a eukaryotic cell and a prokaryotic cell. Absolutely. Well, I shouldn't even say that. Never mind. That doesn't hold true. But a plant cell, right? That's what you're thinking of, is our plant cell versus our animal cell. But bipedalism, standing on 2 feet, that's relatively unusual, right? We have a large brain size. Our cerebrum, the thinking part of our brain, is larger compared to our body size than other animals. And we also have complex language. Those are all human traits. Stop me as I go along, because there's no reason for me to go through all these. Some of these you haven't seen either. Let me see if yeah, and our biological organization, from the single cell all the way up to the biosphere. If you look in your book, it actually goes farther, doesn't it? You can see it starts with individual atoms, but these are not living. This is a cell, and that's the first living of these.
Do they have to be living to be a level?
Not of organization, not of organization, but if I was asking you, depending on how I worded the question, I would not put this in there. Even though these make up cells, these themselves aren't living. It's the cell that's the smallest living component. So we have organs, and we'll do more of this. This is a tissue. And in a couple of weeks, we'll be doing that in labs. So we have a cell, but cells of a similar type make up a tissue. And then a variety of tissues makes up an organ, and then an organ system. Let's see. Humans are classified in what domain?
Eukarya.
Eukarya, absolutely. Oh, and this is the one that's a tough one. Which of the following is not a characteristic of living things?
See? Can you hear me? Oh, yeah,
can.
see.
I can. I'm giving everybody a chance to think about it, because this was a tough one in class.
No, because my microphone sometimes doesn't work.
I'm gonna go with A.
In class, we got to that. We got to A and C, but sexual reproduction is not necessary. Sexual reproduction is a function of what happens
Right.
in eukaryotes, right? So eukaryotes, for us, yes, that's how we reproduce. And reproducing is a characteristic of life. It just doesn't have to be sexual reproduction. Bacteria don't reproduce by sexual reproduction. This one, you are right, and that's what a lot of people choose. But that is taking in information, taking in stimulus, and making a change based on it, or making a decision, or adapting to it. And that's
So a change from the environment.
Yep. Yep. This was when we talked about coronavirus. And we talked about scientific reasoning, and we talked about developing hypotheses and them being testable hypotheses, right? We should be good at that now. We got to experiment and observe. So of these, we were able to observe a couple weeks ago what was happening in our potato cell and water and salt. And that allowed us to use that knowledge to develop a hypothesis. And then our hypothesis included this prediction. And we have been able to experiment and observe in our paper, we're not doing this. We're not repeating the tests or modifying our hypothesis. But if we were in a situation where we worked in research or something and something didn't work, we generally, if your life's research is involved in this, you don't just say, oh, bummer, that didn't work. You say, oh, well, if that's the case, then what does that mean? And you look again and you modify your hypothesis and you repeat your tests, and you work toward constantly answering the next question. But we analyzed our results and we're reporting to me your conclusions. So talked about control groups and different types of experimental design, how clinical trials work.
The clinical trials was when we brought up the blind experiment and the double blind, right?
Correct. Yep. When you do where is it? We actually brought it up on this one with experimental design, because when you're actually designing the experiment, you would determine whether it's going to be blind or double blind. I don't know. There has to be occasions that experiments are not blind when they deal with people, but I would imagine that most all of them are considered blind or double blind experiments at this point. But there may be some where there's no way for you to be ignorant of what's happening to you. So there's a whole lot of different types of things out there. Different kinds of experiments. Let's see. Did you guys hear that Johnson & Johnson, their vaccine has been approved for emergency use? So we now have 3 vaccines out there.
Wait, with there being 3 different vaccines, how similar are there? Like, why are there 3?
Because there are varieties of ways and mechanisms to make a vaccine. So Pfizer and Moderna actually were pursuing a newer type of vaccine, but not unheard of, using messenger RNA. And that's why both of those need to stay frozen and to be frozen at lower than normal freezer temperatures, like -70 degrees C. And that's because they're made of messenger RNA, which degrades very easily. The Johnson & Johnson is a more common type of vaccine. And that means that they're giving you a kind of a piece of the coronavirus, and then they're having you react to it. Messenger RNA is used to have you make a protein that helps your immune system react. So it's just a little bit different approach. But we're going to find that like even Moderna and Pfizer, they're different companies. They didn't coordinate on it. It's just how these different companies went about it and which approach they took when they started is why they're different and made differently now.
When you get the vaccine, do you know which one you're getting?
Yep. There's minor differences. One, Pfizer and Moderna are the only 2 that have been out so far. This week, Johnson & Johnson will come out. So you knew you had Pfizer or Moderna if you've already been vaccinated. And depending on which one you got, the second shot is a week different. So there are different types of mRNA, or I think different types, there's probably slightly different sequences that they used. I'm guessing. So one, you get your second shot in 3 weeks, one, you get your second shot in 4 weeks. So, and I believe they will tell you. I know when I signed up for one, it had said it was a Moderna vaccine. So, all right. That's not a good clicker question for right now. Does anybody need to go through the basic chemistry? Well, we'll find out. We'll find out how these go and we'll see.
The only question I do have on chemistry is when the electrons are where it's like -1 or +2 next to the electrons,
Mhm.
I believe, and what affects how many electrons there actually are compared to like protons.
Okay. So actually, let's work our way up to that then. So the atomic number is equal to the number of how many? A number of what?
Protons.
Protons, right? B. And the atomic mass is equal to the number of
B.
B, protons and neutrons. And so isotopes are differing numbers of neutrons, and we didn't deal much with those. There's not really much to do with those from a practical standpoint for us. But when we talk about an ion, and that's what you're talking about with the numbers, if it's got a +1, remember that as we look at our periodic table, let me go back. I think I've got a closer one. There we go. So as we look at a periodic table, the number that's shown above, so nitrogen here, N, the number above it is 7. That tells you how many protons there are in nitrogen. That will never change. But as this appears on the periodic table, nitrogen also will have 7 electrons. None of these are ions. As they show up here, none of them are ions. So that means this would be a plain N, and it would have 7 protons and 7 electrons. So they offset each other. So it is a neutral, right? 7 positives, 7 negatives. But if that were to gain an electron, electrons are a negative charge. So now it would have 7 positive charges and 8 negative charges. So it would be a -1.
So if there are more protons than electrons, it's a plus. And if there are more electrons than protons, it's a minus.
Exactly.
But depending on how much, like if we're missing 1 electron, then it's a +1. And if we're missing 2 protons, then it's a -2.
Okay, you can't miss protons though, right? Or
so
you
there's
can.
just like more electrons. There's 2 more electrons than the number of protons.
Then it's going to be -2.
Got it.
Right. Number of protons will never change. So it's always going to be electrons going up or down. Now, why up here? First of all, remember what the numbers on top are. That's how many valence electrons are there. So our nitrogen has 7 electrons total, but it has 5 valence electrons. How many valence electrons does it want to be happy? It wants 8, and it's only got 5. So if it has an opportunity to pick up up to 3 electrons, it's going to be happier.
Why does it want 8?
Because this is full. This is a full shell. 8 electrons, with the exception of this and this, right? Because our very first shell only holds 2 electrons. All of these across here have between 1 and 8 electrons in their outer shell. So if I look at oxygen here, it has 6 electrons in its outer shell. I know that because of this number. And it always wants to have a full shell. The outer shell, an element always, I'm acting like it's thinking, it's not thinking, but it's always going to strive to have a full outer shell. Since this has already got 6 electrons, it is more likely to gain 2 electrons to get 8 electrons in its outer shell than it is to lose 6 electrons. And then its outer shell is smaller, but it's still full. So you can tell by looking at this side versus this side, whether something would rather gain electrons to make a full shell or lose electrons to go down and fill it, to get rid of its outer shell that's not full and go down 1 shell lower. Okay. I know that can be confusing.
It makes sense.
Okay.
Right.
Oh, we already did that one, right? Let's
Yes.
see. Let's see what else we've got here. So we talked about bonds. Oh, and there was the ions practice. So we know that this one has 1 extra electron. This one has lost 2 electrons. This has lost 1 electron, lost 2 electrons, gained 2 electrons, right? We good with those?
Yes.
And then this way, we were able to see how many protons, neutrons, and electrons are present in anything. Okay, so this showed us, now we could do this on here. If we found S, it's right here, that has 32 as its atomic number, or atomic mass, not atomic number, atomic mass. So there's atomic mass, there's the element itself, and then this is its ionization. So it has gained 2 electrons. Because it's number 16 down here, I know it has 16 protons. I know it started out as 16 electrons, but because it's, I also can see that it's gained 2. So I have 18 electrons. And then 32 minus 16 is going to give me my number of neutrons. So I have 16. So I could figure all of those out if I was given a periodic table, or I'd need to give you the periodic table plus these as well, right? All right, so clicker question. Determine the element that contains 20 protons, 18 electrons, and 20 neutrons. What are you going to start with on this?
20 protons.
20 protons, right? Because the 20 protons is going to identify that element for us. And so based on that, we can immediately get rid of the potassiums, the K. So it's one of these two. And
Well, it's B.
Mhm.
it's what?
B.
It's, I seem to like B, don't I? I'm not gonna have to switch that. It seems like I've done a lot of,
Either B or C.
yeah, I don't know what, I don't know.
Maybe
Luckily,
you don't want, you feel like the first one will be too obvious and the last one will also maybe be too obvious.
yeah, that's exactly the way I feel when I set it up. Just so you know, that's arbitrarily decided on in my exams. I don't pick it, it comes up randomly. So you
think
can't
that's
go
a
by
safer
that in
bet.
the exam. All right, so it is B because this is the atomic mass. And the fact that it's 40 means that I've got 20 protons and 20 neutrons. All right. Covalent bonding is sharing electrons. And that's exactly what, like here is a carbon atom. And our carbon atom has, if we look back, our carbon is here and it has 4 electrons in its outer shell, right? I can tell that. There's my 4 electrons in its outer shell. Carbon is really handy this way. And that means that it can actually bond with a lot of different things at once. That's why it's so valuable to us in all of our organic molecules. So in this case, each hydrogen has 1 electron. So if they share, 4 different hydrogen atoms can share with this 1 carbon molecule. That's methane. Okay, that's the structure of methane. Hydrogen, remember, only has 1 electron in its outer shell. So if it shares with somebody else, they share it, then they both have full outer shells. And that's what you're seeing here, is just how that works, those sharing of electrons. So that would be a covalent bond. The other bond that we looked at is an ionic bond. An ionic bond really is a 2-step bond, because the first thing that has to happen is we have to have 2 different ions, 1 positive and 1 negative. And in order to do that, for example, here's sodium. Sodium has that wimpy 1 electron in its outer shell. It doesn't want that. It'd rather get rid of that, or I guess I should say the likelihood is better that it can get rid of this 1 electron in its outer shell than it can gain 7 electrons. So we've also got chloride, who has 7 electrons and wants the 1. They're not even going to bother to share. They don't want to share. This sodium would rather get rid of it because it doesn't want all the other electrons. It just wants to dump this 1. So the first step in an ionic bond is to give away an electron to something else. That means whatever's giving it away is positive, whatever's taking it in is negative. And now positive and negative attract, so these 2 come together. That is not as strong as a covalent bond. Okay, sharing makes them stronger, but either way, opposites attract, and this is a bond. The last one is a hydrogen bond, and a hydrogen bond is a very weak bond. It's an important bond, but it's a weak bond compared to either the covalent or the ionic bond. So as you look at this, our hydrogen bond forms usually, it almost is like an ionic bond, because the hydrogen bond is forming between charges. Positive and negative are attracting, but this is not, let me do this again. Whoop, I think I did this last time. Okay, so this is water. Here's oxygen and here's hydrogen. And because hydrogen and oxygen have a covalent bond, meaning they're sharing electrons, the oxygen actually pulls harder. Okay, that's called electronegativity. So it pulls harder on the electrons. That means the electrons spend most of their time right around the oxygen atom instead of around the hydrogen atom. So the hydrogen, for all practical purposes, is losing its electron. That means over here on this part of the molecule, it's got a more positive charge. It's not an ion. It's not a complete charge. It's not a complete positive. It hasn't lost its electron, but it is more positive. Now, because the oxygen has pulled that electron close to itself, it is more negative. those partial positive and negative charges are what make make hydrogen bonds. Okay, so when you see a hydrogen bond, it will be between an oxygen atom and a hydrogen atom, or a nitrogen atom and a hydrogen atom. We didn't look at that, and you don't need to worry about it. But that's when you see hydrogen bonding. And we know the power of hydrogen bonding because of the characteristics of water. We know that if you fill a glass, you can fill it over the top, right? And it doesn't go down the side, it holds on for a few seconds. You can actually fill it all the way up and it goes up over the top and holds there. And that's hydrogen bonding. Those pieces are holding together. Hydrogen bonds are present in water, but they're also present in DNA. So they hold our two strands of DNA together. And that's really important. But if those were stronger bonds, we wouldn't be able to separate it and we wouldn't be able to make more DNA, which we needed to do to survive, to make more of us, to make us taller and bigger and make new skin cells. Let's see. Oh, that's all. We've talked about that. I have that. We talked about solutions, right? What about pH? How are you guys with pH?
Yes, would be good to be going over it, but also I had a question on the solutions. When the salt, I think when the salt goes into the water and it becomes a solution, were there like different titles of what the salt was and what the water was before?
Yep. Remember, solute is going to be what dissolves. So solute can be, solute could be salt, solute could be sugar, solute could be, it could be amino acids are solute. Anything like that that will dissolve in the water or another solvent. We use water as our solvent for almost everything. But solvents, there are other solvents as well. But so yes, solute goes to solvent to make solution.
So the solute is what dissolves in the solvent.
Correct. All right, and pH. pH. Do we get the practicalities of pH? Right? So 7 is considered neutral. What is 10 considered?
Also a neutral?
10?
Basic.
I would consider that basic or alkaline. In general, you give a little wiggle room on each side of 7 that you'd consider as pretty much neutral. But as soon as you start getting up to 9, 10, 11 and higher, I would, if I was asked, I would say that those are definitely basic. Same with the lower end. I would say 6 or below, I would call acidic. I wouldn't ask you, I'd be obvious on one side or the other what they are. I won't ask you like a 7.5, what you consider that. But how do I tell the difference? What's the difference between a pH of 10 and a pH of 12? And that's going to be the percentage of hydrogen ion concentration, or of hydrogen ions. So hydrogen ions are always higher down low. So the highest amount of hydrogen ions is at a 0 or 1, like in there. And the hydrogen ion concentration goes down as we move up. And each one of these is 10 times. So even though a pH of 9, I would call basic, it has 10 times more hydrogen ions than a pH of 10. Okay, even though they're basic, the highest amount of hydrogen ions are down here at the bottom in the acidic range, and the lowest amount are at the top. But each time there is a 10-fold change in the amount of hydrogen ions. As I go up, the hydrogen ion concentration is going down. As I move down, the hydrogen ion concentration is going up. I think, yeah, here we go. Okay. So if a solution has a pH of 6, it is considered
Acidic.
acidic, and it has more or less hydrogen ions than a pH of 8.
More.
It does, it has more. And a solution that has a pH of 7 is,
Neutral.
it is neutral. So a solution with pH 10, so up at pH 10, has 10 times less hydrogen ions than a sample of pH
4 or
9.
9. Are you
so
following
do
that,
they
Katie?
go by increments of 10? Kind of.
The hydrogen ion concentration goes by increments of 10. So every whole number is 10 more or 10 less, 10 times more or 10 times less hydrogen ions. You just have to determine which direction you're going. So if we're still at a pH of 10, then it has 100 times more hydrogen ions than a solution of pH. We have a pH 10, and 10 has 100 times more hydrogen ions than a solution of pH
1.
Let's go back here for a second. So these are our numbers. And I think what you're doing, I think you're trying to make like 1 to 10, but you're right here. And 9 has 10 times more hydrogen ions than pH 10. 8 has 100 times more hydrogen ions than pH 10. You see what I mean? You're going by whole. You're going by whole. I don't know if my dog heard whoever's dog on there or something here, but it certainly bothered her or them. So each round number, Katie, is going to is what you're looking at. So if pH 9, we know number 1, pH 9 is basic or alkaline, but we also know that pH 10 has less hydrogen ions and pH 8 has more hydrogen ions.
Is that the case as well with acidic?
Yep. So let's look at pH 2. pH 2 has 10 times less hydrogen ions than pH 1. And it has 10 times more pH ions or hydrogen ions than pH 3.
So does then 3 have oh my goodness, 100 less than 1? 100 ions
Yes,
less than 1?
100 times. 100 times less than 1.
And then 4 has how many less than 1?
This would be 10, 100, 1,000.
Okay. So we're basically adding a 0.
Yeah, that's a good way to put it.
Okay. I think I get it. now.
Okay. So then, if we're starting at pH 10, which of our pHs does 10 have 100 times more hydrogen ions?
8,
Other way,
Oh, 12.
12.
12, right?
Yes.
You gotta remember that down is always lots of hydrogen ions, up is less.
So the closer to neutral or 7, you have more ions. And then the farther away you get from neutral, the less ions you have. Or at least for the acidic, I'm guessing. Is it the opposite for basic?
No, you're actually right for the basic. As you go away from neutral, you're gonna have less and less hydrogen ions.
Okay.
But at neutral, any of the acidic range has more hydrogen ions.
Okay. Need to write that down somewhere.
Take a picture of this. So a pH of 5 has
I totally forgot.
1,000 times fewer hydrogen ions than a pH of 2, but it still has 100 times more hydrogen ions than pH of 7.
Okay. Yes.
I'm late. I totally forgot.
All right. So, so buffers, we talked about one specific buffering system that we use in the body, and that's the bicarbonate buffering system. And we talked about hydrogen ions, remember, is how we determine pH. And remember, the more hydrogen ions that we have, the lower the pH, the more acidic, right, Katie? The more hydrogen ions, the more acidic.
Yes.
All right. So this is a way that we can bind hydrogen ions so that we don't have free hydrogen ions. So if our body is producing a lot of hydrogen ions, we're able to balance that by moving this, by combining. Notice these are all the same thing. They have all the same elements, right? And they have them all in the same numbers. The difference is we recombine them depending on the circumstances in the body, and that allows us to control the hydrogen ion concentration. That allows us to control homeostasis of pH. That allows us to control our pH in our body. Let's see. Oh, this is a good one. We know dehydration synthesis and hydrolysis, right? Yes, of course we know this. This one is this dehydration synthesis or hydrolysis?
Which one?
The one you're seeing the same thing. You're just seeing a close-up of it.
Hydrolysis? Did I say that right?
The word's right.
Dehydration.
So it's dehydration.
It's dehydration synthesis. How do I know? Because these were attached to my monomers, right? And now they're not. I've removed a water molecule. If I remove a water molecule, that's dehydration synthesis or condensation. Let's see, I know I have another one here that shows hydrolysis. Let me see. Well, this isn't quite, there we go. This one shows hydrolysis.
And with that, are you adding something?
You're adding, this is an enzyme, but you need to have water added. So that water that you took out to make the molecule, you've got to put it back in to separate the molecule. And that's hydrolysis, lysis using the water molecule.
So wait, why are we adding the molecule for hydrolysis?
So that when you break these apart, they're complete monomers. You're adding back the molecules that you had taken away in order to connect them. Now you want them to be a complete glucose and galactose, so you've got to add back that water molecule. The other thing is, remember that hydrolysis does not require energy, but dehydration, synthesis, and condensation do. Remember, that's building up muscle versus sitting on the couch. Oh, here we have some clicker questions. When sodium bicarbonate is combined with water, the sodium bicarbonate is called the solvent. True or false?
False.
False.
Correct. What is it called?
Solute.
Solute. And what are our 4 organic compounds that we talked about? I'll give you a hint. With proteins, right? What are the other ones?
Is this where carbon comes in?
Carbohydrates.
Carbohydrates,
Lipids.
lipids, and what? It's the last one. It was kind of an oddball for us. The rest of them we think about because we eat them. What
Is
makes
it-
DNA?
Aminos?
Nucleic acids, right? DNA and RNA. And what are the monomer of our nucleic acids? Guess that's a good follow-up question since we're not remembering our nucleic acids. What did we make up our nucleic acids? What do we make up DNA and RNA with?
Aminos, right?
Nope.
Oh.
But what, Taylor, what do we use amino acids for? To make up
Proteins.
proteins, right? So our monomers for proteins are amino acids. Our monomers for carbohydrates are
Just the monosaccharides.
it is monosaccharides, and our lipids don't go together that way. There's not a monomer polymer relationship. But our nucleic acids are made up of nucleotides.
Oh, okay.
Remember, nucleotides. Whereas our lipids are, if we're talking about triglycerides,
Oh.
right? Remember triglycerides, we have 3 fatty acids in a glycerol molecule.
I have another one I gotta get to.
Okay, bye, Taylor.
Thank you.
Actually, I'm going to stop here shortly because I've got a meeting as well coming up. So are there any specific questions before we stop?
The Four Kingdoms, are we gonna be, any of
I'm not worried. I don't think I'm worried about the Four Kingdoms, but the domains, I want you to know.
that? Okay.
Katie,
I
do you have a
just
question?
came in.
Katie?
Yes, I did. So this is more of like so I have accommodations and I was filling out my purple sheet,
Uh-huh.
about to email it to you, but I don't know what to put for, my person emailed me. The test is the same time as class on Wednesday, correct?
Yes.
Okay.
Do you have a class right after, or do you want to take
No,
it?
it's my last one, so that still works.
Okay. All right.
Can you just kind of just say what I need to study for, because I missed out so much. What are the topics? Just a few hints.
There is 185 or so, some
Oh.
slides. Yeah, I didn't really give hints. We were just going through.
I see.
So, yeah, I really, I can't do it that way. Sorry, Zambia. I just don't.
Oh,
even
no,
know.
I was just wondering because I was late. I was working on my paper and I emailed you actually, if you don't mind checking it out later and get back to me about just a question about the paper.
Okay.
All right.
So yeah, if you guys have, I'll take like one or two more specific questions, but then I've got to go too.
Oh, so there's no I just came in. I was just wondering if the so the review session isn't gonna go until 3:30 like it was planned, right?
It wasn't planned till 3:30. It
Oh,
was planned till 2:30.
oh, okay. I didn't realize. I thought the office hour was.
Well, it is, but
Okay.
I wasn't planning on spending 2 hours doing it. So
Oh, I get it. Okay.
yeah, yeah.
Thanks. I'll
But
I'll have
I'll
to
get
watch the
this.
recording then.
I'll get this out as soon as I can this afternoon, Dylan.
Okay.
I've got
Thank
a
you.
meeting and then I'll try right after that.
I'll make sure to watch it before the exam. I'll try to.
Okay.
Last question. The
Yep.
paper is due next Wednesday or Monday?
What paper?
The
Are you talking about the osmosis paper?
I believe so. Yes. I think that's
It's
the only
due the day that what, are you a Thursday lab or a Tuesday lab?
Tuesday. So it's due Tuesday.
All right, but it will be due next Tuesday before lab for you, right?
Yes.
You haven't done it yet. You haven't done the experiment yet.
I have not, no.
Okay.
I did do the pre-lab, but I had to check to make sure that it was good or okay.
I checked some. When did I check? Yeah, tonight they're due. I've tried to keep up on them, but I have not checked since yesterday morning. So
I turned it in technically the Monday or Tuesday of last week.
okay, then I've looked it over and sent it back.
Okay. Thank you. Have fun at your meeting.
Thank you.
All
All right, Dylan.
Have a good day.
Thank you. You too. I will see you later.
You too.
Bye.